Thanks for putting together this amazing conference. I have to apologize that I haven't asked too many questions in real time, but the time difference with California and having a 1-month-old and a 3-year-old to tend to has made it kind of hard. But anyway, even when I was holding a kid or something like that, I had my little headset and I was listening to everybody. So it's been great. It's also fun because Stefano and I were at a conference. We were riding a car together to Heathrow in February, and we said, see you next month in Brussels. And here we are. So it's been great to see old friends and get to know new people here. All right. So the work I'm going to talk to you about today is the result of a really cool collaboration with a bunch of friends, both at Berkeley and outside. It's been spearheaded in my lab by Megan and Nick with the help of Emma, Simone, and Armando. But it's also the result of work together with the lab of Chris Wiggins and Bache, his student, Mike Eisen and his postdoc, Mike Stadler and grad student Jenna, and Xavier Darzac and his postdocs, Thomas and Mustafa. And Alex already introduced the sort of question that we're interested in, which is to try to understand how you go from a single cell to a multicellular organism. And we asked this in the context of the fruit fly Drosophila melanogaster. She gave you a little bit of a sense that at this stage, all of these nuclei that went through multiple rounds of division already know what they're going to be when they grow up, even though they look the same. And the reason they know what their cellular fate is because they are already expressing a unique combination of transcription factors. And years of genetics have revealed that these transcription factors are organized in a cascade with a first layer of activators and repressors expressing shallow gradients that leads to genes expressed in broad domains, that gives you genes in 7 stripes, that gives you genes in 14 stripes, that ultimately segments the embryo, and think of your vertebrae as your segments, and also assigns those segments a molecular identity. A cellular identity or an identity, sorry. Meaning, are you going to be a wing or a leg or an eye? Okay. We don't only know the molecular identities of these transcription factors, we also know the regulatory connections. Okay. So this is an amazing state of the art to start asking these sorts of questions in. And yet I have a complaint, and this is maybe more appreciated also by the locals over there. But just like this is not a pipe, this is not a fly. And what I mean by that is, if I change the names in this network and I give it to the world's experts on flies, they would have a hard time telling me that this gives you a fly. You cannot calculate a fly out of this network description. And what I'm going to try to convince you today is that part of the reason you cannot do that is because these network descriptions are missing information about space, time, and concentration. So what we're trying to do in my lab is to endow each one of these arrows with molecular and quantitative information that allows us to reach a predictive understanding of developmental decision-making. Basically, the question is, could I predict the developmental phenotype from this network topology? And so just to be more specific, what we're after is predicting output patterns of gene expression from knowledge of the input distribution of activators and repressors and their distribution of binding sites on the DNA. Or to put this in other words, we want to be able to calculate how DNA sequence prescribes developmental input-output functions, basically the functional relationship between output levels of gene expression and input transcription factor concentration. Now, you might say, well, this is crazy. You're like, there's just too much stuff going on. You're never going to be able to reach this sort of understanding. And all I can tell you is that for the one data point I have, which is the work we did in bacteria when I was a PhD student with Rob Phillips at Caltech, we got it to work. So this is just an example from bacteria where like repressor doesn't let polymerase bind to the DNA. These are theoretical predictions for the level of gene expression as a function of the number of repressors, where each curve corresponds to a different regulatory parameter, such as binding energy and number of promoters. And the point is that all these curves existed before we took the data. These are 0 parameter fits, if you want to call them like that. And then we went on to take the data, and this is what we did. So we showed that to a very large degree, we can predict gene expression from just knowledge of the DNA sequence in bacteria. Even more cool, I would say, all these curves look like very different, look like kind of adventures in and of themselves. But it turns out that you can collapse all this data onto a master curve once you reveal the natural variable of the system, which happens to be a repressor fugacity, which is something like mapping a non-ideal gas onto an ideal gas. So I don't know, at least for bacteria, it looks like we can make this work if we spend 10, 15 years working on it. And of course, now you could say, well, but eukaryotic cells are more complex and development is even harder. So And I agree with that, but the question is, what are the molecular details that I actually need to keep in order to pull this off in an animal? Okay, so I agree it's more complex, but which ones are the ones that I need to account for? To do that, one of the first things you need to realize is that when I plot the simple input-output scheme, there's a lot more stuff going on under the hood, right? There's, you got your input transcription factor pattern that dictates the concentration of your transcription factor in the nucleus. That has to bind to the DNA. That has to dictate the transcriptional activity of an individual promoter in a single cell. And ultimately, many, many cells doing this together figure out or realize the microscopic pattern of gene expression that we usually observe, okay? So the talk today then is going to be about trying to figure out how does each one of these steps work as a means to be able to write governing equations of developmental decision making. And basically what we need to do is, as I said, endow these little cartoons with a little bit more molecular, but also importantly quantitative and predictive information. And to do that, I'm going to follow this approach that is very much inspired by this great article by Joel Cohen, which is Mathematics is Biology's Next Microscope, Only Better. Biology is Mathematics Next Physics, Only Better. So the types of questions I like to ask is, can physics be biology's next microscope? What can I learn when I use theory and thinking about a biological process that I couldn't even learn with the fanciest super resolution microscope? And obviously this is not foreign to, this is very familiar to what we have seen throughout the talks over the last 3 days. But yeah, so basically to put it in other words, why can I learn about biological systems when, as Darwin says, I use this extra sense that math gives me? So I'm going to divide the talk in three short parts, and please interrupt me along the way if you have questions. I don't need to make it all the way to the end. First, I'm going to show you a sort of computational and experimental microscope that we developed in order to reveal that transcriptional activity at the single cell level is bursty. Then we're going to put on our theorist hat to, very much like inspired by the types of model that Alex introduced you to, try to understand how can you realize this control of transcription through the modulation of bursting at the molecular level. And that's going to lead to predictions about what is happening actually at the promoter. How does the binding of transcription factors at the promoter dictate transcriptional activity? And that's going to lead to a very cool technology that we're just in the process of developing that I claim should be able to give us allow us to do a whole in vivo biochemical dissection of the transcriptional processes at the promoter in real time in a living embryo. All right, so the first case study I'm going to use is probably the most understood pattern of gene expression in all of development, which is this little stripe of expression of the even skip gene, okay? And initially the stripe is produced by two activators, Bicoid and Hunchback, and you see that leads to a broad domain of activation. And then repressors come along and they restrict the expression domain of the stripe, which results in a stripe of around 6% of the embryo length, okay? So this is all nice, but what you need to realize is that what I'm showing you here is the averaged amount of RNA produced throughout in that area, right? So this is giving you no information about what's actually happening at the single cell level. So the first question we wanted to ask is, okay, like we are used to seeing these patterns, how do single cells work together to give you this pattern? And to do that, we used the MS2 system that Alex already alluded to. We developed this in my postdoctoral lab in Thomas Gregor's lab at Princeton, but also was developed in parallel by Alex's collaborator, Natalie Dostadny at the Curie, and it uses a technology that was invented by Rob Singer a few decades ago now. And the idea is the following. We're going to add a sequence to the gene of interest that, when transcribed, forms a loop. The loop is recognized by a protein fused to GFP, such that every nucleus in your embryo is going to have a little spot of fluorescence, and that fluorescence is going to be proportional to how many polymerase molecules are actively transcribing the gene. This is what it looks like when you put it all together. In red, you see the DNA, and in green, the RNA signal. And I just want you to notice how you start with a broad domain of activation, and as development progresses, you're going to see how that domain of activation gets restricted and restricted into the little stripe that we've seen multiple times in situ experiments. There you go. There's the stripe. And the one fascinating aspect about this experiment that I never get tired of pointing out is that you're basically staring at an in vivo single molecule experiment, because each one of these dots reports on the transcriptional decisions made by an individual promoter in a living embryo, okay? So if you want to figure out what the single cells are doing, all you need to do then is to zoom into one of these cells and plot the number of polymerase molecules actively transcribing as a function of time. And what you see is that this number of polymerase molecules is modulated in a punctuated fashion, okay? And in particular, if you zoom in, you often see these little trapezoidal functional forms, okay? So what do they tell you? What do these trapezoids tell you? Well, the thing to remember is that what our technology is measuring is the number of polymerases that are transcribing, but presumably what transcription factors do is regulate the rate at which you load polymerases onto the gene. These two quantities are not the same, but they're very much related. And they can be related by a very simple model where you say that initially your gene is in the off state, and then you turn it on, you start loading polymerases, so your fluorescence grows linearly. If you wait all the way, and if you wait until the first polymerase makes it all the way to the end and falls off, but you keep loading polymerases on the other side, now you're going to have a constant fluorescence level. And finally, if you turn off the promoter, you're going to start losing polymerases at the same rate that you loaded them. What I'm trying to tell you is that the simplest model to explain these trapezoids is a model where the promoter is on only transiently, okay? And these are these so-called transcriptional bursts. And by no means are we the first ones to observe them. People have seen them in bacteria and yeast, in mammalian cells, in other animals. So bursts seem to be the rule more than the exception in biology. And the simplest theoretical model onto which to project bursting is the so-called two-state model, okay? Where we're going to posit that the promoter fluctuates between being in a transcriptional active and inactive state, stochastically with some rates, and only when the active state, it transcribes at a rate R. So if you can measure the rate of polymerase loading as a function of time, you would see these little bursts where each parameter of the model defines the burst frequency, the burst duration, or the burst amplitude. So now that we know that cells regulate or cells transcribe in a burst-like fashion, how do you reconcile what single cells are doing and the accumulated amount of RNA that you see in these patterns that corresponds to the mean rate of transcription? So you can solve this model in steady state. It's pretty simple. And what that model tells you is that the mean rate of transcription is dictated by this combination of parameters, where you have a term that tells you what the fraction of time you spend in the on state, a term that tells you how long, at what rate you transcribe when you're in the on state. But what this tells you is that if you want to create a stripe of expression, you can do so in three different ways. You can do so by increasing K on, or increasing the burst frequency, decreasing K off, or increasing the burst duration, or increasing the rate of initiation, or increasing the burst amplitude. And any of these mechanisms, any of these strategies, or any combination thereof can give you a stripe. So now the question becomes not just how do single cells transcribe, but given that single cells transcribe in a burst-like fashion, how do they modulate bursting in order to create patterns of gene expression, okay? And to answer that question, what we need to do is measure K on, K off, and R, and measure it as I move along the embryo. And this would be trivial, completely trivial, if I could measure the rate of polymerase loading as a function of time. Then I would measure the burst size, frequency, and duration, and I would be done. The problem is that the promoter state is hidden from my view. And what I see is the number of polymerase molecules that are loaded onto the gene. So these two quantities, again, are related. So I could, in principle, make an inference. Yet this is easier said than done. Just to give you a sense, in 2011, we developed this technology. And it wasn't until 2018 that in collaboration with Chris Wiggins, we figured out how to do this inference. So for almost a decade, we were sitting on terabytes and terabytes of data that we had no clue how to analyze. And so how do we do it? I'm not going to go through the math, but let me just walk you through just the general idea. Again, we want to infer the hidden promoter state from my observable fluorescence. And if you look at this, you could argue, well, this looks a lot like when I try to infer the state of an ion channel, is it open or closed, from the current recordings that I can get from, let's say, patch clamp single molecule experiments. And to do this inference, typically people use these hidden Markov models that are super powerful, but they cannot quite be applied to our situation. And the reason they cannot be applied is because, as you can see here in the ion channel case, there is a direct correspondence between the state of the channel and the current. But here, in the case of transcription, the same state of the promoter can lead to two different fluorescence levels. And the reason for this is because a polymerase stays on the gene for a given amount of time, given by the elongation rate and the elongation time. And that leads to a memory. And regular garden variety hidden Markov models couldn't deal with memory. So we had to rewrite them. It's fun because, well, it's interesting because now it takes like a week to analyze the data, whereas the data takes 3 hours to acquire. But now we can do this. And let me show you what I mean by we can do this. Here I'm showing you the individual trace of a cell. And this is the fluorescence, the number of polymerase molecules. And you would be hard pressed to tell me when a burst begins, when a burst ends. but now we can infer the most likely state of the promoter with our model. Okay, so we can infer when the promoter is on and off. We can do this for every nucleus across the embryo. And here I'm color coding those nuclei following whether the model tells us that it's most likely to be on or off. And what you can see is that even in the middle of the stripe, where you have the highest level of expression, the promoters are bursting, they're going on and off. There's no such thing as being on all the time. Now that I can do this, I can move along the embryo and ask what are the values, the magnitudes of the different parameters of the two-state model that dictates bursting, okay? For example, we can plot k on the burst frequency as a function of the position, and you see that this magnitude, the burst frequency, is modulated in a step-like fashion, which tells you that some of the transcription factors that regulate the stripe are talking to k on. If you plot the burst duration, interestingly, it remains constant throughout the embryo, okay? Which tells you that none of the 4 transcription factors talks to k off. Finally, if you plot the rate of loading as a function of the position, you see that this is also modulated in a burst-like fashion, which tells you basically that the transcription factors, some of them talk to k on, some of them talk to r, maybe some of them talk to both. This is more or less consistent with what people have seen all over the place. Sometimes people see only burst frequency being regulated, sometimes they see a combination of parameters, sometimes they see burst duration. And in general, we've gotten really good at, or relatively good at, inferring which bursting parameters are being regulated, okay? The one thing we still have very little idea about is how is this regulated, implemented at the molecular level, okay? So I'm saying, in some sense, I'm saying that we need to move away from how does a single cell activity lead to the transcriptional output, and start asking, how does, for example, transcription factor binding, or the action of transcription factors, dictate transcriptional bursting, which ultimately leads to the formation of patterns, okay? For example, I mean, you could imagine two different scenarios where transcription factors need to be bound throughout the duration of the burst, or one where transcription factors only are bound at the beginning of the burst, right? So how can you figure this out? And obviously, what this is calling for is some sort of correlation between binding and transcriptional activity. So to start making progress in that direction, what we decided to do is think about this from the theoretical perspective. And the simplest model you can think of, and this is a stupid model, especially after you saw Alex's talk, but let's entertain this for a second. Let's assume that the off state corresponds to an activator site being empty, and the on state corresponds to the activator site being occupied, okay? In this very simple model, you predict that the burst frequency is given by the activator binding rate, meaning that the more activator I have, the higher the burst frequency. You also predict that the burst duration is independent of activator concentration because it only depends on the residence time of the activator, which can be modulated if you, for example, had control over the binding affinity, okay? So let's say that you believe this model, then the prediction that you just made is one where, for example, the timescales of activator binding and unbinding should be the same as the timescales of bursting. And if you want to test this, you need to measure binding and unbinding in vivo. And Alex already alluded to these measurements. So this is data that we took together with Xavier Darzac using the super cool Lattice light sheet microscope, which by any measure you can think of is like 100 or 1,000 times better than a confocal, okay? So this is now, all the other ones were like microscopes in air quotes. This is an actual fancy microscope. And we looked at Bicoid, one of the activators of the stripe. What you see here is a nucleus of the fly. There's another nucleus here, another one here, another one here. What you see here is a little Bicoid molecule that stayed on the DNA, that was bound on the DNA, and because it was bound there, it stayed there, and you could see it with a flash. And what you see now is multiple flashes of Bicoid binding and unbinding on the DNA, okay? And yeah, hopefully you can see it. It's like the frame rate is not as good when you do it through some. Anyway, what we can do then is ask how long does Bicoid stay bound on the DNA on average? And you get something that looks like an exponential distribution with a residence time of about 1 second. And this is all great if it wasn't for the fact that the bursts that we observe last for 5 minutes. So there's some sort of disconnect here between the timescales of binding and the timescales of bursting. And this disconnect seems to be widespread. So on the top here, I'm showing you basically some summary on this log scale of different bursting dynamics measured in a bunch of different organisms. On the bottom, I'm showing you the timescales of the different molecular processes that you would call the usual suspects for transcriptional control and transcriptional bursting. And what you can see is that for the most part, most of these molecular processes occur on much faster timescales than bursting, okay? So clearly we're missing something here. So what we decided to do is make the model a little bit more complex, and now this is getting even closer to the model that Alex introduced you to. We're going to assume that we have multiple binding sites here. I'm showing you 3, but we do this for 6. And we're going to assume also that the number of activators bound to the DNA dictates the rate of transcription in a linear fashion, okay? So when you put this together, then the other thing we do is we impose the binding and unbinding rate to be on the order of a second to be consistent with our microscopy measurements. And then you simulate the system and you ask, what does the rate of transcription look like as a function of time? And what you see is that this rate of transcription is fluctuating around a given mean, okay? So it's not bimodal, it's not bursting in any way. Like it's not spending a lot of time in a high state or a non-state. This, you know, we were surprised by this, but then we realized, all right, well, we're missing a key element that it's been mentioned here also a few times, which is the fact that here we assume that the activators were binding independently. What happens if you include some sort of protein-protein interactions that, for example, stabilize activators in the presence of another activator on the DNA? When we do this for reasonable values of the activator of cooperativity for reasonable energies, what we get is bursting. And not only that, we get bursting on more or less the right timescales. You can see a few minutes each time, okay? So it looks like even though the activator binds and unbinds by itself on the timescale of 1 second, cooperativity can stabilize binding for long enough that to get bursting, okay? But then, you know, how, you know, this is nice with the simulations, one of these things where the computer understands it, you know, what can we understand what's happening? So what we did is we plotted, we made a measurement of our simulation. So this is simulated data, not actual data. And the blue points tell you the waiting time distribution for the different activator binding events that we saw, okay? What you see is that there are 2 regimes here. There's an early regime that looks exponential, and that's basically the regime that we could capture with our Lattice light sheet microscopy. But there are also a bunch of events that last much longer and that are much more likely to occur than you would expect by a simple exponential distribution. And I think this is very much consistent with what Alex just told us about. You can then ask, well, do these long binding events play a functional role? Well, you know, this is the simulation when I have both short and long binding events. And then you can simulate the deletion experiment where you force activators to unbind after 20 seconds. And what you see is that you lose bursting, okay? So this suggests that bursting is driven by long-lived binding events. But if that's true, then why didn't we see these with the Lattice light sheet microscope? And there are multiple reasons for that. One that was alluded to in the first day, I think, was that GFP bleaches within 3 seconds or 10 seconds or something. So, you know, with GFP alone, you can get to see these guys. But more fundamentally, what we and most people have been doing when we say we are measuring transcription factor binding, is we're saying we're measuring transcription factor binding at a random location throughout the nucleus. And sure, in that location, maybe there's for sure non-specific sites, maybe there's one specific site, a few specific sites, but you don't know what's happening there. The holy grail is to be able to measure binding at the target locus. Not only that, to know that it bound there and to correlate that with the transcriptional activity. Okay, this is something that, you know, many people have been trying in single cells. Some people have had, like Dan Larson and Tineke Lenstra have had great success doing this in yeast. But for the most part, this remains pretty hard, okay? So basically this led then to us rethinking how can we do this measurement? And you know, you need to keep in mind that, you know, there's no super resolution approach that can co-localize a binding event and a transcription site. You know, you're not gonna get this by brute force with super resolution microscopy. That just doesn't work on the timescales that are necessary. So what we decided to do is try to think, well, can we leverage the data that we have both for binding and for bursting? And this led to this, what I would call a data analysis microscope that we call burst trigger averaging, which, you know, I'm super excited. This is super fresh data, so I would love to hear your thoughts. And I really think this is one of the things that is, like, you have these experiments once in a life in your career where you think, well, okay, now I'm going to start doing this. This is one of those experiments, at least worth changing my life for. So let me walk you through the idea here. To do this, I'm going to switch gears. I'm not going to talk about if stripe 2 anymore. I'm going to talk about a different activator system. The details don't matter, but I'm going to talk about snail activated by dorsal. These are expressed along the dorsoventral axis of the embryo, and dorsal is also a gradient along this same axis, and you can see how snail is expressed in a domain along the ventral side of the embryo. Okay, basically the same thing as with Eve, just a slightly simpler pattern. And what we can do now is fuse dorsal to venous, or we can use a fusion that somebody else already made. So we can measure the input and we can measure the output as well with MS2. Okay, so green and magenta shown here. And we can ask, following really cool work from Ido Golding and Jun Ma, and also in part from Ibrahim Sisset, we can ask, do you see what happens when you measure the dorsal venous concentration, the input concentration in the vicinity of the site of nascent transcription? And the answer is, you see crap. You don't see, that's all noisy. There's nothing interesting in there. And this is true wherever you see it. So, I mean, if you looked at this, you could say, well, I don't see any dorsal binding. The cool thing is that once you start averaging over a few snapshots, by 50 snapshots, already you start seeing that on average, there is more dorsal signal at the site of nascent transcript formation than anywhere else at any random location in the nucleus. Okay? And not only that, that signal has some dynamical signatures, or seems to have some dynamical signatures. What do I mean by that? This is dorsal concentration or enrichment at the hunchback locus. Hunchback is a gene that dorsal doesn't interact with or doesn't talk to, okay? This is, for another nucleus, this is dorsal at snail, which is its target gene. And you see that the overall signal, the average is higher, which you would expect, but also there are these little excursions, right, that kind of last on the order of 5 minutes. So, you know, this is only data from one nucleus. It's hard to infer anything interesting from that. But one of the first questions you could ask is, are these excursions, do these fluctuations correspond to binding events? And if so, how do these fluctuations correlate with transcriptional activity? Like, you could imagine a scenario with this is binding and this is when you get your burst, okay? So how do you test this? How do you test this if bursting is a stochastic process, if the whole thing is very noisy? Like, you know, it's hard to extract statistically meaningful information from one trace at a time. What we decided to do is basically very much draw inspiration from super resolution microscopy, from spike trigger averaging in neuroscience. So these are basically ideas where you can spread your signal over some dimension, usually over like many repeats, and then you can average it together to increase your signal to noise dramatically. What we did is the following. We grabbed our MS2 signal, the number of polymerase molecules transcribing the gene, and we inferred the promoter state using our hidden Markov models. At the same time, we have this very noisy signal of dorsal on the locus. And what we do is we grab the dorsal signal and align it according to the start of each transcriptional burst, okay? So you put them all together, like I'm showing you on the right-hand side, and then you take an average, okay? So on the left, what I'm showing you are different transcriptional bursts in black and the corresponding dorsal signal in red. And again, it's hard to see anything in there, but once you start doing this, it only takes it only takes you to average a few bursts to start seeing a signal, okay? So let me show you this to you in a little bit more detail. This is snail. This is a typical transcriptional burst. This is the signal of dorsal at the beginning, during a burst. And this is dorsal at a gene that it doesn't interact with, okay? So what we see is that dorsal surges in concentration right before the burst and then remains high throughout the duration of the burst before going down, okay? So again, as I mentioned, the disclaimer here is that these are super early days, but there seems to be a statistically significant signature. And it makes you wonder whether we actually are seeing binding events that lead to transcriptional control. But obviously the next questions that one needs to ask are, you know, how do these search dynamics dictate bursting dynamics? Like what I mean, for example, does the amplitude of this search in dorsal dictate the amplitude of the transcriptional burst? Does the appearance of the search dictate the burst frequency? So these are questions that with more data, we can start parsing them out and answering them. The other question that you might ask is, how are these searches created? Like, you know, is it just proteins coming all together? Are there clusters of dorsal? We have some ideas, and if you want to, you can ask me during the questions part about this. But, you know, there might be some stuff. We believe that there might be some little clusters that are involved in this, that it's just not single molecules getting to the promoter. But why do I keep saying this is kind of an experiment to change my life for? And it's because I think that we can finally start revealing the anatomy of a burst in vivo, in a living animal. What do you mean by that? We got really good at measuring transcriptional dynamics. We can see these bursts. That's all. That's okay. But now we can correlate this transcriptional dynamics with the binding of activators before, during, and after the burst. You could imagine starting to ask questions such as, well, what do pioneer transcription factors that are in charge of opening chromatin do? Like, do they need to be just there before the burst? Do they need to be there throughout the whole burst, for example? And I claim, and we can talk about this more during the questions too, that we might even be able to get access to what's happening with the chromatin landscape, with the histone modifications, by using some fancy sensors that people have been developing. And again, it would be fun to be able to see histone modification dynamics at these timescales are completely unattainable by genome-wide approaches, and would be very complementary information. But most importantly, and going back to the very motivation of this talk, I claim that if I want to write governing equations for cellular decision-making development, I'm going to have to figure out some of these molecular details. I don't know yet which ones of these are all that relevant, but I need to start being able to ask these sorts of questions if I want to figure out what the governing equations are. So, and I also, I'm kind of preaching to the choir in this audience, but again, I'm very, very amused by this idea of different microscopes. And I know this is just, I was pushing this more for narrative purposes than anything, but basically I showed you how we can use an in vivo microscope to reveal single cell transcription with MS2, then a computational microscope to infer what's happening at the promoter in that individual DNA molecule, then how we can use theory to make predictions that lead to a theory experiment dialogue where we measure transcription, binding of transcription factors, and ultimately that led to some sort of crisis or paradox in terms of binding rates and bursting rates that maybe we're starting to resolve with this new burst trigger averaging, which I would call a data analysis microscope, that allows us for the first time to correlate binding at a specific locus with the transcriptional activity of that locus. All right, so with that, I already acknowledged the people involved. It's an amazing group of people to work with. And I also need to acknowledge my lab. This is some snapshot from a few years ago, and funding sources below that give us the freedom to have fun and do crazy things. And all of you for your attention, and also the organizers for this great workshop. Thank you very much.
Thank you, Hernan. That was a beautiful and clear talk to end the workshop with. Any questions? Stefan, go ahead.
Hey, Hernan, that was absolutely beautiful.
Hey, Stefan.
I was wondering, given all this excitement with condensates, what's your thought on maybe this cooperativity coming from formation of small condensates? Yeah.
No, that's super cool, and it's even more relevant. Some people alluded to this. One of our results from Bicoid is that it doesn't bind uniformly throughout the nucleus. It binds at different. It has higher binding in localized regions, which we call hubs, just to give them some name. And dorsal has something similar. If you stare at it, and if you image it with slightly fancier imaging conditions, what you see is that there are little clusters of higher concentration. Okay, so we're trying to do better at imaging them now, but what you see is that they come about, they sometimes touch the gene, and then they go away. Okay, not only that, if you ask, what is the average distance? What is the distance between clusters and a locus? These clusters seem to be closer on average to target loci than to non-target loci. Okay, so we don't quite know whether this is playing a functional role. And your friends over there at Dresden have also some really cool evidence, for example, for roles of buffering noise and such things. So what we need to do now is to be able to track these clusters and the transcriptional activity to see if there's a functional role of these clusters. But I still don't know how to think about it, but there's interesting stuff here that we're just starting to scratch the surface.
I mean, we've looked at a pioneer factor in an optical tweezer, KLF4, undergoing a kind of a pre-wetting transition. So
Okay.
I should send you this.
Yeah.
We
No. Hold on. This is the bioarchive
yeah.
paper you guys just had. Which one?
Exactly. It's just put it on the bioarchive a couple of weeks ago.
And you know, one of the things that these clusters depend on the pioneer-like transcription factor. These hubs depend on the pioneer transcription factor Zelda, and Zelda seems to make little sausages throughout the nucleus. So there's something funky going on there.
We should talk anyway. I'll drop you a line.
Yeah, absolutely. I would love to for sure. Yeah, I don't know. The picture of a binding site, like a transcription factor coming and binding, I think is not like Eric started his talk showing this nice picture of like a looping enhancer and all of that. And I just think we just don't have the right picture yet.
That's a nice little bridge to the next question, perhaps, because I think Eric wanted to ask something.
Eric.
Not about that, but
no, it's okay.
Hernan, so thanks a lot. That was really great. So I wonder if I can ask about sort of the key picture of your talk towards the end, where you line up the binding at the dorsal locus with the signal that you inferred, right, from the inference of when the promoter is on and off.
Yep. Yeah. 1 second. Yeah. So, keep going. I lost it. There you go.
So the dotted signal, that is so when are we in a burst?
You're in a burst. Basically, we align the bursts according to when they start. Okay. So but there are issues. So, you know, in principle, there should be a little line, like, you know, a square, but because we don't have that much data, we need to incorporate some burst that had another burst like beforehand. And that's why the whole signal is a little bit rough. And so we're working on that. It's just that it's a lot of data. Sorry, I'm answering a question that I don't know if you asked.
No. And then the dorsal signal at the hunchback promoter. So that should, in principle, that's a control. So why does it still show quite a bit of up and down, do you think? Is it just noise or? Well,
by ChIP-seq, dorsal does bind to hunchback. It just doesn't seem to play any functional role. So part of the question is
snail transcription, right? I mean, there's no reason for it to co-organize in time with snail transcription.
Sorry, no. It's just that this one we're aligning according to bursts of hunchback activity. Even though I'm not showing you the black line. Okay, I see what you're saying.
Okay.
Yeah. So, you know, we have different controls. One is at the hunchback locus, one is at a random site on the DNA, though you can do the burst trigger averaging if you don't have an actual transcription site. So we need to play with different targets. Like, you know,
all right.
You know, here, let me show you one more thing that just to show you how it's unclear that there's something wrong here. So this is, for example, Bicoid binding at hunchback. And it does bind to it, and you see an enrichment. But check it out, Bicoid binding at snail is it's not enriched, it's de-enriched. It's depleted. So I don't know what's happening. I think there's more to unpack in this whole business.
All right, very good. Thank you.
Sure.
Any other questions?
So then I will follow up. So do you think that
go ahead, yeah.
Clusters of dorsal, you think these are enhancers of where dorsal is bound that are flopping around?
No, I don't think. Well, I mean, they could be some other enhancer. I don't think this corresponds to the enhancer of this gene. They're kind of far away, and I don't think the numbers would make a lot of sense. Maybe they are. I should think about that. For sure, people like Justin Crocker have seen that multiple enhancers tend to come together also in organized regions. So maybe there's a little bit of that.
So it's more like maybe it's both what Stefan suggested and enhancers, right? They might.
Who knows? Yeah, no, I agree. So, you know, we're starting to do things where we delete some enhancers, but, you know, there's multiple enhancers for dorsal in the genome. So we'll have to be smart about it. You know, one idea would be to start labeling them with something that labels the DNA independently of transcriptional activity, and then try to see, you know, do they tend to co-localize with those enhancers and such.
All right, very good, thanks.
Sure.
Right. Well, thank you, Hernan.